We left off, we were talking about the immune system. So as I said, we're going to talk about disease-causing microorganisms. And to be able to understand them, we need to understand the environment that they're living in, which ends up being you. And where we left off, we were talking about some of the major players in your immune system. We'll go through kind of the roles that they have and how they interact. And as I said, your immune system can be broken up into two main pieces: the innate immune system, which is a more slower acting process, but it's also a very non-specific general type defense that you have, which includes your skin, which is just a barrier to actual cells that will actively move through and destroy bacteria and viruses that do enter you. The main one being these macrophages that have these very generalistic receptors that if they bump into a bacterium or a virus, they'll take it in, they will digest it, and spit out the waste. There's also these cells called dendritic cells that are scouts. They're out there monitoring. They're very important in monitoring the environment that is you, checking out how you're doing, as well as anything that may be popping in for a visit, whether it's a nice bacterium or whether it's a parasitic one. And we'll talk more about those in a minute. So these kind of get grouped into the innate immune system. The dendritic cells, these scouts, also get incorporated into the adaptive immune response, which is a very specific part of your immune system. With the scouts being important out there, kind of monitoring what's there, they will go back to cells in your immune system that we call the lymphocytes. They'll communicate, talk to them, and activate them and target them and help them, those cells go and cause a very specific directed attack towards whatever bacteria or virus, or in many cases poisons and things that happen to be in your body. The lymphocytes, which we generally call the white blood cells, the ones we'll talk about are the B cells, which help recognize foreign material, T cells, which recognize these foreign materials as well, and also help regulate the response of the adaptive immune system. And then finally, we'll talk a little bit about these killer cells, which help take out your own host cells that happen to be infected by an internal parasite. So each one of these we'll go through, and we will you'll see their roles as they pop up as we go through. Yes?
What are the cells in the innate immune system?
What?
The cells of the innate immune system.
Those?
Yeah.
So it's mostly just the macrophages that, which the closest thing that you can think of them as is like an amoeba. They're very similar to those. They're not an actual amoeba, but they have their qualities are very similar. You can see that they kind of ooze around through cytoplasmic streaming, moving around. It allows them to move. As we'll see, they have the ability to, they're in all your tissues, but they also have the ability to move through your tissue, pop out, travel through your circulatory system to other areas to help out in responses when it's time to fight. So we'll go through that in a second. So as I said, the last thing that we're talking about is that when, say, you do become infected by some, say, bacterium, parasitic, pathogenic bacteria or virus, in order for your immune system to be able to function in a very efficient way, cells have to talk to one another. And communication is very important. The other thing is that they have to first, they have to be able to detect that there's a foreign organism inside you as a host organism, and then it has to then communicate. So if cells, like a macrophage, happens to bump into a bacterium, it recognizes it as a bacterium. It can detect that. It can say that this has a signal or has a signature that's not like any signature in the host organism. So it's able to bind to it, bring it in through phagocytosis, eat this organism, and as we'll see later, it's able to communicate with other cells because because it's now digested something that's not you. It does trigger other types of internal changes in that organism. One of the things that it does is it takes pieces of that organism it just ate and starts presenting it to other cells to say, I found this, I found some type of bacterium inside of us here. Why don't you, if you can, I recognize this, why don't you run with this and tell other parts of the immune system that we've got this problem. And not only do they communicate that there is something wrong, but also they can send out signals to help direct where this particular infection is localized. So organisms, the other parts of the immune system, will move to that area and fight the immune system, not just throughout the body, but in the particular location. So the main way that they do this for detection is through these external receptors. So this would just be in their plasma membrane. They'll have a receptor on the outside. As I said, phagocytes or macrophages have generalistic ones. So they're able to bind to a lot of different signatures, a lot of different signals that they could bump into that are kind of generalistically that a lot of bacteria are going to have. So if an organism has one of these macrophages has these external receptors, it bumps into a bacterium and it binds to it. It's now bound and it's recognizing an external bacterium to itself, and it will take it in and it'll eat it. So anything that it bumps into that it will bind to with the receptors, we call an antigen, because an antigen is just any type of signal, any type of compound inside your body that results in an immune response. So that means, and that's a wide variety of things. It doesn't have to be just a bacterium. It doesn't just have to be a virus. It can be any type of foreign organic material. Pollen can cause an immune response. There's a lot of different, if you cut yourself and get some organic material inside of you, your immune system will wipe that out as well. Even if it's dead organic material, it will still be dealt with. So these receptors are for binding any type of signal that is not you. They can also have internal receptors pointing in. So since they're facing inside the cytoplasm, if something happens to bump into one of these receptors, which is again for detecting anything that's not you, not the host, it will then know that not only it's not that they bumped into a foreign object, that foreign object, whatever it is inside the host cell. So bacteria, viruses, certain strains, well, all viruses and some bacteria do end up moving inside host cells, living inside those host cells for a period of their life cycle. So having these internal receptors facing in helps that host cell recognize that it's infected. It can tell other cells, part of the immune system, that they're infected and that they need to be dealt with. Usually that means just killing them, but it allows for finding bacteria or pathogens external to cells, as well as finding if there's any type of bacteria or viruses that have now moved internal into a host cell. And there's a variety of receptors. They have a whole slew of them. There's general ones and there's specific ones that are out there. So again, this is just to show you what I said about antigens. So in these receptors, they will bind anything that gets into your body that results or triggers an immune response is called an antigen. Now the thing is, antigens can be extremely large, like bacterium, like a virus, like pollen, but these receptors only recognize a very small piece of that overall antigen. Different receptors, some are more general, some are more specific, but only the region that actually ends up binding to that receptor is what's important, and that's what we call the epitope. We can highlight that. That is what that receptor is particularly binding to. Now, one single antigen can have multiple epitopes, or it can have one. Just like a bacterium can have many different proteins, all of them can be recognized and by different receptors, and it's only just small pieces of those proteins that they have that it's being recognized. So antigens can be very large, they can be very small. But only the one little piece of an antigen that actually binds to the receptor is what's important, and that's what we call the epitope. Yes?
Suppose that the antigen is part of an organism.
Yes?
The organism mutates, so the epitope is different. And does that mean that the antigen will be detected?
Yes, and that happens all the time. So there's plenty of microorganisms that evolve very quickly, and that allows them to stay ahead of your own immune system. So we'll talk a little bit about that later, but the AIDS virus is a good example of that. The way that your cells communicate with one another is through the production of these small proteins or peptides called cytokines. And there's a family of them. It's basically the language that the cells communicate in. Different ones tell cells different things. The cells have receptors for these cytokines or cytokines, whichever way you want to say it. And this allows your own immune system to be able to communicate with itself and to be able to get different signals. A lot of times this is for if there is an infection in one part of the body, cells in that area will produce cytokines that will then go throughout the body, which will trigger other cells to know that there is an infection, and that they will then move towards that particular site. What they end up doing is following the concentration of the cytokines. As it goes up to a certain concentration and reaches a certain threshold, they know they're in that area, and thus they will mount a response in that area. So and as we'll see, there's other ones that are there that trigger a number of things. Sometimes cytokines are sent out to tell a particular cell that it's time to die or pass away. There's a number of cytokines out there that are used to communicate from one cell to another, as I said, attracting cells to a site, triggering particular cell responses, and also to trigger more whole organism responses like inflammation or fever or those type of things. So let's talk about inflammation. So inflammation is a very common thing. I'm sure like everyone who's cut themselves has experienced inflammation to a certain degree. Here's your skin. Here's the dermis. Here's the dead cells at the top of the epidermis. Here's a blood vessel running just underneath. These are capillary vessels running away from this blood vessel. And these are macrophages that are situated in the tissue of the dermis. Now, inflammation, the main goal of inflammation is to contain a damaged area. So everyone has had their skin disrupted. You've had a wound. So the thing is that as soon as you get any type of wound on your body where your tissue has been disrupted, inflammation will take hold immediately. That is a natural response to having your tissue disrupted. The cells recognize that has been disrupted, they've been disconnected, and the cytokines are sent out to the area, and it does mount a inflammation response. So let's go through that process. Let's do that. Say that you're walking along and it's not like your interest, but say like you're spending time with some very nefarious folks and you get stabbed. So it happens. And as soon as that happens, this is a very nice one. This is not, see, it's a very tiny knife, and it is a very tiny cut, so it's not that big of a deal. You have friends that have those little tiny model token knives that they collect for, I don't know, D&D fans or Lord of the Rings fans. You've got Legolas knife, something like that big.
It looks more like fortunately, it looks more like one of those things you make with one of those added machines. No, maybe sunglasses for a fly. I
have no idea.
Sunglasses for a fly.
Oh. I'm going to have to look that up because I have no idea what that is. But anyway, you've been cut, and an inflammation response is going to occur. And what that means is you're going to get a swelling in that area. And it can just be triggered. And usually, like when you get inflammation, you get swelling and it gets warm. Typically, that's what you're going to feel. Now, the thing is, like as I said, like you can get just by you being cut, you're going to get an inflammation response. You can also get an inflammation response if you have bacteria or some type of foreign material in your tissue, in your live tissue. Both of those will mount a response. And as I said, the main reason for this is to kind of cordon off and shut off this area. When you get a wound and you open it up, there's some bonuses to having to kind of trying to cordon off that area. One is to prevent blood loss, but the other one is to prevent any bacteria that's in that wound from spreading to other nearby tissue. So the whole purpose of, and we'll go through the actual process of what happens, but it's to basically prevent anything that's now in this cut any bacteria that's in there to go from this area to spread to this other tissue. Because remember, as we said, there's a lot of opportunistic bacteria right on the on your skin, on the dermis, that can't do anything because it's too dry and it's too salty for them to be able to survive. There's also bacteria on the knife because your friend's not, cleaning his knife very well, and those are going to get placed inside of there. So there's going to be bacteria in your wound. There's bacteria all over the place. There's no way for you to get cut and not to get bacteria into your wound. It's just impossible. So that's going to be in there, and the purpose, the main thing that's going to have to happen is there any way your body can just block those bacteria from moving? Because if they get into capillaries and you get into this blood vessel, they can start spreading throughout your body, and then you get sepsis, and you can start suffering really, really bad things. Even if it's not a parasite, it could be what we call an opportunistic pathogen that you've opened the door to them. They're going to take advantage of it until they die. So what happens first? During inflammation, the wound, or the cells in this wound recognize that there has been some disruption. So signals go out, and the first thing that happens is there's going to be a change. The blood vessel is going to become dilated. Not the capillaries, but the blood vessel. So the blood vessel is going to get bigger. It's going to dilate out. It's going to get bigger. And what that does is it's going to compress the dermis, the tissue around here. And what that does is it shuts off. It causes the capillaries to collapse. So with the capillaries collapsing, there's a couple of things that happen. One, some of the small roads are now shut off. The bacteria can't use those to move to other areas. The other thing is that the blood, which is bringing nutrients to that area, it's as you're bleeding and so forth, that's bringing nutrients. It's also bringing oxygen. Those are things that these bacteria need. So by dilating the blood vessel, it blocks the ability of those bacteria to easily move to other tissue, and it also deprives them of a lot of nutrients and a lot of oxygen. So, and of course, it depends on how deep the wound is and all these other things, but generally that's the purpose of it. So the other thing that happens, I think that's in this one, is the blood vessel has gotten bigger and there's more blood flow going into that area. This is one of the reasons why you feel it get warmer, is because there's more blood flowing to this area. Just underneath it. The capillaries are shut off, so the immediate vicinity is not getting any blood to it, but just nearby you are getting a fair amount of blood flow. And in this, these cells that have recognized they've been wounded are sending out cytokines, and they're going out and they're telling the rest of the body that there's a wound here, there's a potential infection, and that it's localized in this area. So these cytokines are flowing out to the blood and going out to the rest of the body. And so the highest concentration is right here, then it dilutes as it goes further away. This white blood cell got that signal, jumped out. It was like one of these macrophages that were in this area. Jumped out, maybe from the liver, who knows where it came from, but it jumped out and got into the circulatory system and started flowing around through the entire body until it got to this location where the cytokine signal was high enough that it changed and moved across the blood vessel membrane and into the tissue, and then added another macrophage to fight these organisms off. Now the thing is, the dilation does, it compresses the dermis nearby, shutting off the capillaries. You get more blood flow moving through, and then on top of it, because it's dilated, that the outer tissue of that membrane has become a little more porous. So it makes it easier for these macrophages to move across and into that tissue. So what you're going to do is you're going to get a collection of macrophages in this area. The number of macrophages is going to go up. As we learned, macrophages are your soldiers. They'll bump into a bacterium, they just eat it. So they're there and they're eating and they're eating and they're eating. But the thing is, they're also dying. So they're dying, they're eating, they're dying. They're in a battle, they're fighting. There's casualties on both sides. Depending on the wound, you're going to start, as these macrophages are moving in, you're going to get a collection of them. And what does this turn into?
Pus.
Turns into pus. So the more pus you have, the more white blood cells have moved into that area and have been trying to fight off these bacteria. Some will live, some will die. But depending on the wound, you'll get either a lot of collection of pus or just a little. But having pus means that at least your immune system's working. Yes?
So I have a question regarding things like like lead or other metals that actually get into your body. So, for example, let's say you're a child and you actually stab yourself with a pencil. It's like it's jamming there. How come our body doesn't use those?
Well, it doesn't. Those, from what I understand, those don't revolve around these type of systems. Like macrophages aren't going to eat up your lead for you. There are different chelators and so forth that do bind to certain things. Lead is really not used by the human body, so there are like certain things where they'll collect in different areas. They can react or like cause types of things. I'm not an expert on lead poisoning, so I don't really know exactly how the human body like really interacts with lead and so forth. But there are like a number of metals, mercury, and a number of things that once inside of you, they do interact with certain tissues. I don't know the exact chemistry of that, but there's not like macrophages or things that are binding to that. They focus on proteins, organic type molecules that are in your body.
So it's mostly like organics, right?
What's that?
It's mostly organics?
Yes.
Okay.
So that's what the receptors are binding to, are different sugars, proteins, lipids, things like that they're going to bind to, because it's a protein receptor that's going to be interacting with it. So it can still do poisons if the poisons are organic. Then they can react with it. Snake venom, things like that, your immune system will respond to. So, all right. So the macrophages are doing their job. This is all this innate immune response at this point. So the macrophages are doing their job. They're very generalistic. The inflammation is an innate response, just to kind of cordon it off. It doesn't matter what's causing it. It's just trying to shut down the system or prevent whatever's in there from spreading. As I said, the white blood cells, the macrophages are going to start collecting, and then you also will get platelets will be coming in to clog up certain areas. So you'll get blood clots forming to shut down and to block the ability of these bacteria to move into your circulatory system and make it easier to go further into other areas to be able to get more nutrition. So going and trying to move between tissue is more difficult, but when you have like your wonderful highway system that is your circulatory system, getting into one of those makes it easier to spread, get to new territory, more nutrients, and then they can make more offspring and make your life that much worse. So all of this is to shut off and prevent these bacteria from moving through collapsing the capillaries, to clogging them with blood clots, to allow moving more macrophages into that area to be able to fight them off. Obviously, it's much more of an issue like when you cut into a blood vessel and you're bleeding out a lot more. Then that's harder to stop. You can't just rely upon inflammation to shut off one of your major arteries through inflammation. Then you got to get a tourniquet and all those other things. But this isn't that class I'm talking about. We're talking about the microbiology. All right, so as I said, just to kind of go through phagocytosis again, macrophages, these are the ones that are just out there eating up bacteria and viruses if they bump into them, as long as they recognize them. As I said, they're sitting throughout your body. Their main roles are to eat, to phagocytize bacteria and viruses. They also eat you, but dead cells of you. So as your cells die, you don't need to be collecting all of those. You don't have, all of your cells are constantly rotating over and recycling. So those old dead cells need to be processed. That's where the macrophages come in. That's one of the main reasons they're throughout all your tissue. Is that your tissue dies, those get eaten up, processed, and reabsorbed by yourself. As I said, they also destroy foreign invaders like the bacteria and the viruses. And as I said, just as I was describing before with the inflammation, they can be recruited to an infection site through, as we learned before, chemotaxis, by following those cytokines. As they go to the higher concentration, they know they're in the area, and then they'll move across the blood vessel wall and into the nearby tissue. Just as a reminder of phagocytosis, this is the overall process. Here's a macrophage bumping into a bacterium. The receptor recognizes it, bumps into a particular epitope on that bacterium, whether it's a protein or a glycolipid or whatever it is. It recognizes a particular signal, at which point pseudopodia will come out. It will engulf the bacterium. It will bring it inside and put it into what we call a phagosome. So this is phagocytosis. So it's just a vesicle, since the plasma membrane reached out, fused on the backside, brought it inside. Now that bacterium is surrounded by an envelope that came from the plasma membrane. So this is what we call a vesicle, is the general term for it. But since it is has a bacterium in it and it's fated to be digested, we call this a phagosome. The phagosome will then fuse with a lysosome. Lysosomes are generated at the Golgi body. Those are just another vesicle full of enzymes, and it's acidic. That lysosome, it has lysozyme in it, and that lysosome is going to fuse with the phagosome, and you can guess what it's going to be called after they fuse. Yes, it's the phagolysosome. And that is now a vesicle. Now that they've fused, you have the bacteria in there. You also have all the enzymes and the acid in there, and that's going to start breaking down and digesting the bacterium. Bacterium will get digested. Any nutrition will be reabsorbed by the macrophage, and then any material that it can't use will be expelled and dumped out. And as I said, with the macrophages, because they did bump into something that they recognize as a foreign entity, they will keep some of that behind. They will keep a little bit of it, and they will present it to other cells to let them know that they found some type of foreign object. They don't know what it is, but they will show them epitopes. They'll show those epitopes to these other cells to let them recognize, to let them mount a response to these particular signals. So this is, and we'll see how that plays a role in with T cells and B cells and dendritic cells in a minute. Now, another thing that your body does is it can produce these things called membrane attack complexes, or MACs. This is still part of the innate immune response. And it's just a cell lysis mechanism. As you can see here, it just looks like a donut, like a protein donut. And it's composed of a number of proteins that are in a circle. It spans across the cell membrane and just creates a hole in between. Here's a electromicrograph of it showing you the holes in this membrane. And by just creating those holes, it just pokes a hole in. Bacteria, and this only functions on bacteria. So at that point, it causes the cytoplasm to kind of spill out and other material to spill in. The bacterium can't control its own membrane and the movement across it. So at that point, the bacterium ends up quickly lysing after that point. So this is just something, and we'll come back to MACs because it's an important part of another type of immune system. But this is a very general type of thing that can be done. If you've got a bacterium inside of you, your immune system does have the ability to produce these MAC proteins to bind to them and then induce lysis. So macrophages are eating them. These proteins are binding to the membranes and poking holes in them. But again, at least for most parts, it's part of the innate immune system. But there's some complexity to that, which we'll get to in a minute. Another thing your body does is it creates a fever. So fever is just where we have like a localized inflammation. You can think of a fever as a full body inflammation. So with when you have a localized inflammation, you do get a localized increase in temperature. Now there's a benefit to increasing temperature of your body, and that's all your fever is that your overall body temp goes up. Doesn't go up too high, but it goes up, well hopefully it doesn't go up too high, but it goes up a few degrees. And the main thing is that any of these pathogens, bacteria that are in you that are trying to take advantage of you, they've evolved with you. Their optimal growth temperature is your body temperature. So if you change that, that's going to have a huge effect on these microorganisms that are trying to eat you and live inside of you. Now, here's a growth curve, standard growth curve for any type of microorganism. As we saw, there is an optimal growth temperature, and below that, their growth rate will drop, and above that, their growth rate will drop. But the thing is that when it comes to growth rate, it's not an even curve. You can drop the temperature on a microorganism, and you have to get really, really cold before they die. But if you just move the temperature up a little bit, you see that it drops off a lot faster. So you don't have to increase the temperature much to push bacteria outside of their optimal growth conditions. And that's why you end up using a fever, because you as an individual can tolerate those few degrees increase in temperature, and you will start taking damage, but not as much as these bacteria. The bacteria will grow slower and give your own body an advantage to killing them off before they become too numerous. So an overall fever, having a fever throughout your entire body, heats you all up. The entire environment these organisms live in is now too hot. They grow slower. You're taking damage, but you're able to kind of run faster than them and be able to kill them before they end up killing you. It doesn't always happen out that way, but that's the general mechanism of a fever.
Question.
Yes?
Does that work with viruses as well? Or is it a little bit more complicated?
No, it is more complicated. Bacteria are more sensitive to that. There are certain viruses that are going to be more susceptible to it than other ones, but it's more universal across all the bacteria. Because like with viruses, they're in your own cells and they're taking advantage of your cellular systems. So it's not, and but there are some viruses you do get fevers with, but a lot of them you don't. But a lot of those times that can just be due to your own immune system reacting to a full body infection. It's just a natural response to it. And even sometimes, like if your body temperature gets too high, you will start accumulating damages. So a lot of times, if certain individuals have a very strong fever and you're at the hospital, or even if you're not at the hospital, sometimes you'll try to someone will try to cool you down, put you in an ice bath to prevent you from overheating yourself, because you do start suffering brain damage after a certain increase in temperature. I think it's like 104. Around there. I know 106 you start suffering brain damage, but I can't remember. Anyone know? 105? 105 is the number? All right, so now we'll move into the adaptive immune system. So this is the one that's targeted. This is a system that's meant to target very specifically a particular bacterium or virus or poison that's in your body. There are two parts of the adaptive immune system. There's what we call a humoral immunity and cellular immunity. Humoral refers to when the adaptive immune response targets an entity, a bacterium or virus, that's external to the host cell. So this is if they're flowing through your circulatory system, they're in between your tissue. This is different than cellular immunity that will attack the actual host cells that are infected. So the big difference is that, as we'll see, with this bacterium that's floating in between cells, all of those epitopes, the antigens and the epitopes, the signal that they have, other cells in your immune system can interact with that. They can bind to those signals. Once that particular organism gets inside of one of your cells, those signals aren't easily detected anymore. A macrophage is not going to necessarily bump into this host cell and recognize any type of those epitopes that are now hidden underneath that plasma membrane of your own cell. So they have to be treated differently. They are recognized, both are recognized, but through slightly different parts of the overall adaptive immune system. All right, just some definitions. For the adaptive immune system, there's what's called a primary infection, and then there's a secondary infection. During a primary infection is the first time you've ever got that disease. So you get it for the first time, your adaptive immune system is going to respond to it, but it ends up responding very slowly. It takes time to ramp up the immune system to be able to target it towards whatever this disease is that it's seeing for the first time. Now, say you win that round, all of that virus or bacterium is now wiped out from your body. Your adaptive immune system can remember the particular signal or fingerprint of that particular disease And then the second time you get it, during the secondary infection, the immune response will go out and attack again, but this time, since it's remembered it, it will be able to mount a much stronger response and be able to attack it very quickly. And many times during a secondary infection, you don't even know you're sick. Your immune system, your adaptive immune system, is able to mount a response so strong and so rapid that particular organ, parasite or disease-causing agent is wiped out before you even know it. With the primary infection, you know it. You're sick, you've got symptoms, it's horrible, and then you recover. The second time, a lot of times you don't even know it. Sometimes it's just like, oh, I kind of feel like my throat's a little swollen, and then it's gone. So there's a lot of time, so we're going to go through the process of primary infection and a little bit of the secondary infection. All right, so it all has to do, again, with this idea of antigens and epitopes, where antigens are anything that causes an immune response, and the epitope being the particular signal. So they are two separate things, but they are parts of the same whole. All right, so in the adaptive immune system, one of the important cells that are part of it is called a B cell. This is a B cell there, and what's these red things on the outside are receptors. These are external receptors that they have that will bind to particular epitopes that happen to come into the body. Now the thing is that your body doesn't have a catalog of all the different bacteria and viruses that are on this planet. That's impossible to be able to have. You do collect a library, as we'll see over time, as you become infected and recover from them. So the thing is that you actually have a collection of B cells in your body. As B cells are produced, each one is slightly different from the other one. Well, there's going to be some redundancy, but there's a lot of variation. As they're produced, randomly, you get differences in the B cells. And the differences are always on these receptors. The B cell itself is the same. The receptors are slightly different. And it's just a protein receptor that's sticking out there. And the thing is that there's mutations that occur at the end of these receptors where they bind things. And it's just a random process. Based on the structure of these receptors, you can have between 10 to the 8th to 10 to the 15th possible variations. That means there's 10 to the 8th to 10 to the 15th possible different epitopes that these B cells can recognize. Problem with that is there's only 4 to 10 to the 13th cells in the human body. You are not made up of 100% B cells. So you only have a small collection of the overall diversity of B cells that there can be. So at any given time, you only have a subset. So you do have a variety, but you don't have every single possibility. So and that just says there's very few cells in the body that recognize the antigen. So, and that's the other thing. For the amount of B cells that you have and the extensive diversity that there is, not every B cell is going to recognize every antigen that enters your body. They are specific, so there's only going to be a small subset that, if they bump into this particular antigen with a particular epitope, they will bind to it and recognize it as a foreign entity. So this is one of the main reasons why the primary immune response is slow, because of this variety. Variety allows for the recognition of a lot of different types of potential diseases that enter your body. But to maintain that diversity, there's only going to be a few in your body at any given point that can recognize it. And that more have to be made as soon as the infection occurs. So the receptor itself, you may recognize this. The B cell receptor, if you took it off the B cell, it's called an antibody. So this is just a figure of the antibody. It has two major mirrored pieces. There's a long chain and there's a light chain. This is all just protein chains. And there's the exact same long chain and light chain on the other side. The region up at the top, so an epitope will bind here, or an epitope will bind there. And this region up here is called the FAB region. This is also known as the variable region. That's the part that mutates. That's why it has a slightly different color up there. That's the part that's constantly changing as a B cell gets made. This is where the mutations occur, and this is where the variation is. Below that, we have what's called the FC region. This is the conserved region. It's the exact same sequence for every single antibody, every single B cell receptor has the exact same sequence down at the bottom in this Fc region, or the stem region, whatever you want to call it. But that stem region binds into the membrane of the B cell, and the variable region is what's sticking out. So let's go to see what happens when a B cell bumps into a particular epitope it can recognize. So here's a bunch of antigens that are out there. There's a green one, a purple one, a blue one, and a red one. Now this B cell has receptors, and if it bumps into this green one, it won't recognize it. That, as I said, these receptors are specific. So if it bumps into it, lands right there, won't recognize it. Bumps into this purple one, will not recognize it. Bumps into this blue one, won't recognize it. There's probably other B cells that will, but not this one. This one, receptor, will bind to this particular red one right there. So let's do that. It bumps in, binds to it. Now this B cell knows that it's got something at the end of it is an antigen. Thus, it's going to mount. This is going to start the process of triggering an immune response. It's bound to it. It bound to just a small region of it that we call the epitope, but that's what's bound to it. That's just part of that overall antigen has been recognized. That happens. It's going to be brought inside, brings it in through endocytosis, and then it's going to grind it up. It's going to break it up into smaller pieces. So as I said, each antigen is made up of many different potential epitopes, and it's the epitope that's important. So this B cell has recognized part of that antigen, brought it in, ground it up into multiple different pieces, and what it's going to do is it's now going to communicate with other cells and let them know that it found something, some foreign entity. And how it does that is through these other different membrane-type receptors. These are called major histocompatibility complex 2. There's one, we'll get to one in a minute. So what these are, these have different, these are proteins, again, these protein type receptors. They have multiple different components. There's alpha and beta subunits. But the important thing with this is that these MHCs, or major histocompatibility complexes, will take, if we go back, you notice this one's broken up into 4 different epitopes. It's going to place all the different epitopes that it just ground up, and it's going to place it on different major histocompatibility complexes. So all of them are being presented. Not just the one it recognized, not just the one it bound to, but all of them. Because all of them, if one part of it is a foreign object, the rest of it is too. So all of them are going to get presented out there on this. So this is how they communicate with another cell. Another cell can now come up, bump into this B cell. The B cell's agitating. B cell's like, I found something. Someone please talk to me. So, and how they're going to talk to them is by placing it on this major histocompatibility complex, put the epitope out there. They're going to see if another cell is going to come along, bump into them, and basically confirm what they found. So here's the B cell. Let's go move forward. Here's the B cell. B cell's sitting there. It's got all the different epitopes laid out there. So the cell that it wants to talk to is what's called a T cell, particularly a helper T cell. The helper T cell has its own receptor. It's a little bit different than the one here. It's basically just half of that. And here's a T cell that's come along. It's got its receptor on there, and it's going to bump into it. And the thing is that this one, as you can tell by its color-coded, this particular helper T cell does not recognize what's being presented to it on this B cell. The B cell knows it has something, but it needs to find a T cell that will agree with it. This is part of the double check process that goes on in your immune system. So the thing is that if the B cell thinks it's found something, it's going to start bumping into T cells. This T cell will bump into it, doesn't recognize it. The T cell's receptor recognizes not only the epitope, but also part of this major histocompatibility 2 complex. So it knows that it's bumping into a B cell, and the B cell is presenting something to it. So if no T cell recognizes this antigen, so say this B cell found something, it's cruising around for a while, it's bumping into T cell, bumping into T cell, bumping into T cell, doesn't find any T cells that also recognize this epitope. What happens is that after a period of time, the B cell becomes what's called anergic and just ends up dying. because there's always a chance that this B cell ended up picking up not a foreign epitope, but a host epitope. It's always possible. Mistakes are made, even in your immune system, and this can happen. So the thing is that this helps for protection against self. You don't want an immune response targeting host tissue, only targeting foreign tissue. That B cell may have just been a bad B cell, got through the system and bound to like some epitope that's part of a protein on your kidney cells or something. So let's actually look at a situation where the B cell did find something. This is where immune response is going to come about. So let's change it up. Now we've got a new helper T cell. This one will recognize it. So in this case, this T cell bumps in. Its receptor does bind to the epitope that's being presented, and also the major histocompatibility 2 complex. So it recognizes that it's bound to that this is a B cell that's talking to it. So what happens is, and also the thing is that if you'll notice, this was the original epitope that the B cell recognized. This T cell is recognizing a different part of that antigen. And this is why they grind it up, because you can have multiple different types of T cells. There's as much T cell diversity as B cell diversity. So by grinding it up and showing different parts, increases the chances that they're going to bump into a T cell that does recognize one of those pieces. Because if one part is from a bacterium, one epitope is, the rest of them are too. So any of them that can be recognized does not matter. Even better, makes it for a more efficient process. So this T cell, what it does is it's going to sit there and bind. It's going to go, okay, got something. Yes, you're showing me something. It's foreign. I agree too. We both agree on this. This is something that's not part of us. So the T cell is going to release some cytokines to the B cell. And basically, the cytokines are telling the B cell, yes, you got something, do something about it. And the main thing that's going to happen is the B cell is going to change its physiology, and it's going to do a number of things. It's going to become what we call active, and it's going to start to multiply. There will be the production of antibodies, and there will also be memory B cells will be produced. So let's look at that process. So the T cell is just there to confirm that the B cell found something. So the B cell is now active. It's going to make more of them. So again, as I said, there's very few B cells to begin with. Now there's a whole lot more of them. This one is the one that's found something. Now you've gone from hundreds to thousands, if not millions of them. They're going to start cruising around. Some of them are going to do what we call class switching. So not all of them. Some of the other B cells are going to run out there and continue to bump into that epitope, because they need to get confirmation all the time that particular epitope is around. Some of them are going to go and do a class switching. They're going to switch over to what we call a plasma cell. Plasma cells, what they're going to do is just produce a whole bunch of those B cell receptors, but not hold on to them, but release them into the circulatory system. Release them to be spread throughout the body, because those things will bind to anything with that particular epitope. So how do those antibodies work? So with antibody binding, anything with that particular epitope is going to bind to it, whether it's that amoeba, whether it's this bacterium, whether it's a virus, whether it was some poison that's in your body, some type of organic poison, it will bind to it. So it comes in, boom, bind to them. Anywhere where there's that epitope, it will bind to it. So one organism can have many antibodies coating it and surrounding it. One of the main things this does is it can hinder the function of that organism, of that virus, of that bacterium, of that particular toxin, and prevent them from interacting with the environment, binding to another cell, interacting chemically with something else, and it kind of slows them down and prevents them from functioning. The other thing that it's going to do has to do with that Fc region of that antibody that was released. The variable region bound to the epitope, and that Fc region is sticking out the backside, and it's conserved throughout every single antibody, every single B cell. That Fc region is the same. That is used as a flag, and it is a flag for macrophages. The macrophages, if they bump into anything and they have a receptor for that Fc region, if they bump into anything that has that Fc region sticking out, that conserved stem region, they'll eat whatever's on the backside of it. So gobble it up. Eat it if it's a poison, if it's a bacterium, a virus, whatever, it's going to eat whatever's bound to it. So this helps in hindering them, but also targeting these organisms for destruction. The other thing these B cells do is they go through a process called affinity maturation. So the thing is that, as I said, these B cells, there's only a small subset that are in there. And there was a B cell in this case that did bind to a particular epitope. But the thing is it bound to it, but it didn't necessarily bind to it as efficiently as it could. And the thing is, there could be other B cells or other variations of that receptor that they have that binds to it more efficiently. And what I mean by that is that, say that this particular one, the one that's up there at the top, I gave it like two pluses. This one, if this particular B cell bumped into that particular epitope and the receptor hit it exactly right, maybe 50 out of 100 times it would bind to it and bring it in and process it. The other 50 times it would just bounce off because it's specific enough to it, but not extremely specific. So the thing is that when this B cell, when it divides, it also tolerates a high level of mutation in that particular variable region of its antibody or its B cell receptor. And it's a random process. So some of the daughter cells, there's going to be differences in that receptor. Some are going to have a higher affinity, other ones are going to have a lower affinity. The ones with a higher affinity, when they bump into it, maybe this one now, out of 100 times, it binds to it 70 out of 100. 70% of the time it's going to bind to it. And then this one, it's only binding 10% of the time. So as I said, these B cells, they need, and you can imagine, if this B cell goes out there and only bumps like 10% of the time it's binding to it, and then meets another T cell and becomes activated, it's only being activated 10% of the time. This one's now being activated 70% of the time. That means that every time it gets activated, it replicates. So after a short period of time, most of the body is full of these, or one similar to this, and there's not many of the low affinity. So the greater the affinity, the greater chance they're going to bind to an antigen, the greater the chance they're going to become activated, and thus the greater the chance they're going to produce more offspring. So this one binds to one, becomes activated again, produces more daughter cells, more random mutation. Some are not going to be as as specific. Other ones will be even more specific. This one's now binding 98% of the time. So that one is much more efficient. And almost every time it bumps into an antigen, it binds to it, brings it in, becomes activated, and heads off to go bind more. And that also means the antibodies that they're producing for the plasma cells are more specific. They're binding even better. So the efficiency, every single round, the efficiency is going up. The specificity is going up every single time. So after a while, in your immune response, some of the B cells that you have are going to become memory cells. And these memory cells get stored in your lymph system. So memory cells are dormant or relatively dormant. They're just used to remember, thus memory cells. To remember the infection. The infection, you've won, you've defeated this particular group, and now you want to remember it if you ever get this particular organism again. So if the bacterium comes back, you'll have these memory B cells, which are now at a very much higher efficiency, a higher affinity than the original ones were. And you have you have a whole bunch of them. You have thousands of them stored in your system, ready to go, waiting for another infection to occur. So the B cells are there just to remember that particular signature in case you happen to get infected with that virus or bacterium again. So you get a much more, so that's why in the secondary infection, it's a much more rapid, a much stronger response. So let's see that with that. Here's all your memory B cells during the secondary response. Now this is where the dendritic cells come in. They're out there monitoring. And if they monitor and they pick up that signal, they'll come over and present them to these memory B cells and say, look, I found this. And if it is the exact same epitope that was found before, then these B cells become activated. And now we have a whole lot of B cells. They become active, and okay, I've already settled that. They're going to produce. There's the antigen that's being presented to them. they're going to go through the exact same thing. We're going to get production of more B cells, we're going to get production of plasma cells, we're going to get production of antibodies. And the thing is, not only is it a stronger response because we have more cells starting out, because we have all these memory B cells, it's also already gone through maturation, affinity maturation, so the efficiency of it's that much that much better. So it's a much stronger, much faster response, and this is why a lot of times you don't even know you're infected the second time, because the response is just so massive that these things get wiped out before the organisms grow up to such high levels that you actually start showing symptoms. Yes?
Let's say in the case of like the common cold, people get it, they heal from it, but then they get it again.
Well, that's because you don't get the same one. Because we'll talk about the common cold later. The common cold has a high mutation rate, so you get it, you get antibodies for it, it goes around the world, comes back the next year, and it's mutated enough that it's not the same thing anymore. The epitope is different enough that you can't recognize it. And the thing is, you've probably got other versions of the cold that you just never notice and just kind of wiped on through. But every time you do get sick and you know that you got sick and you get the symptoms, that's because you got a new strain that's epitope that's slightly different enough that you don't have the exact same epitope anymore, and the immune response has to be mounted all over again.
Does that also apply to like influenza?
That's a little bit different. We'll talk about influenza. That one is not as much of a mutation. They do mutate, but that one they have an ability to mix with other flu viruses. So, and we'll talk about the flu virus later. But yeah, that's one of the main reasons you get the flu again too. So dendritic cells, as I said, these are the scouts. These are largely used for, a lot of the immune system is about regulation and making sure an immune response is targeted to non-host cells. So the dendritic cells, they're throughout the body as well. They're really great because they're monitoring you and the local environment at the same time. They're very wonderful little cells that are running through. You can see they've got these very long pseudopodia. They can move in between your tissue and grab stuff and bring it back. So they're always checking out, collecting proteins, toxins, antigens, not like full bacteria, but the pieces of them. And there's a lot of pieces to be collected. And not only are they collecting that, they're collecting the pieces of you as well. So they're always out there, just out there scavenging and collecting. They're the hoarders of the of your body. So, and as I said, long pseudopodia to be able to reach all the different crevices in between different parts of different cells. Now, they also have non-specific receptors. So they're in that way, they're like the macrophages. If they bump into anything, they're going to grab it and they're going to bring it in. If they do end up grabbing an epitope that they don't recognize, they do change their physiology a little bit. They do recognize they've picked up something that is, in a sense, non-host. So what they do is, if they're triggered, they do become hyperactive and they start collecting even more. They start grabbing more stuff because the thing is that as I said, there's not a lot of B cells, there's not a lot of T cells out there that could recognize whatever organism's in there. So the more epitopes they have, the better. They're the scavengers. They're the ones that are trying to let everyone know what's out there. So they're trying to get every piece that they can find and then present it to other organisms. And what they do is they take all these in, and kind of similar to like what the B cells did, they take them, they grind them up, and then they present them out on major histocompatibility complexes. In this case, they present it on MHCs 1 and MHCs 2. So, and I'll redefine those in a second. And they also travel to the lymph system to present this to T cells. They also present it to like memory B cells. So they're communicating with the neurons. We'll just talk about the T cells and how that one, how they function with them. So as I said, they present them on two different major histocompatibility complexes. We already know the major histocompatibility 2 complex is recognized by helper T cells. Major histocompatibility 1 complex is recognized by a different group called cytotoxic T cells. We'll talk about them in a minute. So the thing is that why they have these two different things is so that they can communicate with two different groups. And this is what largely separates the humoral response from the cellular response. So this one is for the humoral, for being able to detect things in between tissue. That's where the T cells are running around. Or the helper T cells. This one is for a cellular response to find those particular organisms that are already inside tissue, inside a host organism. And we'll talk about that in a minute. But there are the two different types, and there's slight differences in their overall structure. Well, what first we want to talk about is how they interact with the helper T cells. So we'll go back to that. Here's, now as I said, when they become hyperactive, when they do pick up something, they do change. And one of the things they change is they start producing what's called a co-stimulatory molecule. This is just a way for other cells to know that they're hyperactive, that they've picked up something different. If they haven't, they're still picking up stuff, and it's mainly just you. And this is part of this double check system, that they're picking up parts, and what's usually in these things, what's blue is you, and what's red is not you. But the thing is, the one up top, it's just picking up parts. It just goes out there and grabs. And it's mainly just been picking up what slough off that you've been doing. But it still grabs, it still presents it out there. This is what I'm finding. But this one picked up a new signal. So this one's picked up some stuff, and it's also letting people know that it's hyperactive. So the thing is, this is where the T cells come in. We've got a whole slew of T cells, just like we have a whole slew of B cells. This T cell is going to bump in to this dendritic cell that's not hyperactive. And in this case, this one's got the right receptor. It does recognize it. But what happens is, because it doesn't have the co-stimulatory molecule, this T cell goes, oh, that's me. They're not getting that co-stimulatory molecule. So this one, it kind of lets that T cell know you've slipped through the system. You're a mistake in this whole process. You're actually recognizing host epitopes. So it's best you go off peacefully and die and get removed from the system. So it's a double check in that regard. But if it's got the co-stimulatory molecule and there's another T cell that recognizes it, this one knows that this is an actual foreign entity, and then the T cell goes through a very similar process to what we saw with the B cells. So dendritic cells can help in this process make more T cells that are specific for a particular epitope that's being picked up. So T cells, just like those, they're going to divide, they're going to go out into the body, they're going to bind with other B cells that are picking up similar epitopes. They're going to activate those B cells, and the B cells are going to go off. They're going to multiply. They're going to class switch. They're going to go through affinity maturation. They're going to produce antibodies. They're going to go do all that stuff. So the more T cells and the more B cells that are specific to this particular organism, this particular epitope, the more efficient the system is, the faster it is, and the quicker that this particular parasite is going to be removed. You also get memory T cells being produced as well, and they are important in activating macrophages, which we'll talk about in a minute. So T cells that have been activated can go activate other cells as well. Yes?
What happens to the antibodies that are released, like the receptors? Like in the instance that there's a cold, you catch a cold, or you catch like some sort of like foreign material, you release the receptors, but what happens like after it's been cleaned up?
Well,
Just stay around in the system?
No, well, anytime a macrophage bumps into one, they're going to take it up, even if it doesn't have anything at the end of it. So they'll be scavenged and processed, and probably some will probably get excreted out of you and so forth. So they only have a certain lifespan. They will get removed after a period of time. And just like with any of these, there has to be a constant. There has to be an epitope around for this to be maintained. As soon as that epitope disappears, just like we saw before, the B cell has to bind to it to become activated. If there's none around, then the B cells aren't activated, and the whole thing starts shutting down, and you get fewer and fewer in number over a period of time. So it all gets shut down, and anything else gets reprocessed. So when a T cell becomes activated, as I said, it can go and talk to macrophages. As I said, like macrophages are out there. Macrophages are out eating stuff, and they're presenting epitopes as well. They're like, hey, I've got something. I think I found something. And this T cell, he got activated too, comes over, bumps into this macrophage, is presenting this out on his major histocompatibility 2 complex, recognizes it, same thing. T cell goes, yeah, you've got something, you're good to go, buddy. here's some cytokines, go run off, go do what you got to do. And what the macrophage ends up doing, the macrophage is now excited. It's become activated. One of the things it does, it gets bigger. And one of the ways it becomes more active, it produces more internal toxin. It will start fusing with other macrophages to become massive multicellular macrophages that are just cruising through and eating up stuff. So it's another way to kind of have another kick up, another notch to be able to get these macrophages out there and breaking up and eating things. Because anything that they, if they're in that area and they're picking up those epitopes, then they're in an area that they need to be more active and thus digesting anything that they need to in that area. Okay, so to go back to the dendritic cell, as I said, they presented out on 2 different major histocompatibility 2 complexes. And I'm glad this guy is highly expressive so I can get all the different cells that I need out of him. So the thing is that, as I said, here's one co-stimulatory molecule. There's the major histocompatibility 2 complex. That's going to communicate with the helper T cell. Here's the major histocompatibility 1 complex. As I said, that communicates with different cells, cytotoxic T cells. So cytotoxic T cells will recognize host cells that have been infected by a bacterium or a virus. Now, the dendritic cell, they were out there just grabbing stuff and presenting everything on both of these. So the advantage to that is they didn't know, they just picked up junk in the environment. They don't know if that came from a virus or it came from a bacterium, but it doesn't know where it is now. It doesn't know. It just found it in the environment. And so by presenting it on both of these, it allows for the dendritic cell to trigger a humoral and a cellular response just in case. So both of them are going to get mounted. The T cells are going to run off, or these cytotoxic T cells are going to run off. Now there's a great thing that your cells are constantly doing. So your cells are always monitoring what's inside of it and telling the rest of your body what they're made out of. So there's this process. So there's always anything that it's in your cytoplasm, that material is getting ground up and being presented out on major histocompatibility 1 complexes. And the vast majority in a fine, healthy cell, it's always the signal, the epitope that's being presented is always of you. So the thing is that cytotoxic T cells, if they bump into it, won't recognize it and they'll move on. Those cells are fine. They don't have a signal that the cytotoxic T cell could recognize, so it's just going to move on and bump off of that one. But when your host cell has a particular infection inside of it, a bacterium or a virus inside, it grinds those up just the same. Grinds up itself and grinds these up. It's a very generalistic process, non-specific, and it presents those out as well. So this organism here that's infected, it's grabbing itself and presenting it out on major histocompatibility 1 complexes, but also grinding up these viral parts and putting them out as well. That particular epitope that dendritic cell picked up may have been one of those signals that's being presented by this host cell. So if this cytotoxic T cell bumps into a host cell that has this major histocompatibility 1 complex and has the epitope that it recognizes, it knows that cell is infected. So it will release a different cytokine. This one tells the host cell, you're infected, it's best that you left us and go to the cell heaven or whatever it is. So this cell is now going to go through apoptosis, it's going to die, and it's going to get eaten by macrophages, reprocessed, and thus the virus or the bacterium inside will die as well. So this is the way that they're able to get the cellular response and get them removed, because the helper T cells aren't going to be able to, and the B cells and the antibodies and the helper T cells aren't ever going to, if they're inside of another host cell, they can't interact with them. They can't recognize that's in there. It's being protected by the host. So these cytotoxic T cells are the one way to be able to get around this. So off it goes, toast. Macrophage comes in, eat it. And everyone's happy. Except the bacterium. All right, so there is self-testing that goes on. Some T cells and some B cells do get through. They can recognize host signal. But there is a self-detection mechanism in place. B cells and T cells are tested when they're first produced. They are tested by your own cellular systems they're presented host proteins, host epitopes. And if they react to those in that controlled environment, they're quickly removed. If they don't react, then they're sent off to the rest of the body to be able to interact. B cells, this occurs in the bone. T cells, that occurs in the thymus. Body degrades, as I said, cell components and is constantly presenting it to those cells. If they do react, they're destroyed. And these are, and as I said, that's where they're processed. That's where you get the B and the T cells. Because in the production of these cells, it's a random process. Your body doesn't know, so it's just random generation, testing, and then whoever passes the test gets sent off. Anyone who fails gets sent to the grinder. Good thing college doesn't work in that fashion. All right. Just one last thing before you go. There is another class of cells called the natural killer cells. This will become important in some other certain organisms, some viruses that we'll talk about later. So as I said, with the cytotoxic T cells, they're relying upon the host cells to be processing what's inside of them and presenting it out of major histocompatibility 1 complexes. Now the thing is that there are viruses and bacteria that can subvert that system and block that presentation from occurring. So if a cytotoxic T cell comes up and bumps into it, there's no major histocompatibility 1 complex. So it just bumps and moves on. So the thing is, that's where the natural killer cells come in. If they bump into a host cell that's showing some sign of stress, which usually means it's not showing the proper signals that it should, it will immediately get killed and removed. It doesn't know if it's got a bacterium in there or not. Maybe that cell is precancerous. Maybe there's a number of reasons what's going on. But these cells will just, if there's some sign of generic stress, they'll wipe out and kill that particular host cell and prevent it from replicating or potentially sending out more viruses or bacteria to the environment. So that's the last one of that.